Okay, hello, welcome to another week here in Biogeography. This week we are going to be talking specifically about historical biogeography and how we sort of piece together like some of these things as far as looking at how lineages evolved over time of specific organisms and how biotas evolved, and looking at sort of the evolutionary history on some broader scales, and specifically some of the methods that we use to actually do that. It's kind of worth noting. I'm gonna kind of walk through some of the history of historical biogeography here. So something that's worth pointing out is that historical and ecological biogeography started out mostly as separate branches, but in recent, I guess maybe recent decades, they have been sort of merging their methods and approaches so that they're less distinct. Like some of these are kind of blending together. So they started out sort of separately, looking at separate things, but over time, starting to realize the benefits of sort of working together and merging some of those ideas and approaches to get a more comprehensive understanding of how things have changed over time. So what I want to start out with is sort of looking at how historical biogeography has changed. Some of the major approaches have changed over time. So we'll sort of take a little bit of a tour through how that's shifted over time. In the first, I don't know, maybe not the first, but one of the early sort of ideas or theories in historical biogeography was that it's something called center origin dispersal or center origin theory, where um, basically people thought that the origin of an organism was found, it sort of uh, was found somewhere and then radiated out from it. So basically things dispersed out from like in a circle. So like an organism would show up in evolution and like via evolution, and our species would show up and then radiate out and disperse out from the center. In this case, like sort of somewhere around here. And it's an interesting sort of idea. What we're looking at here is an early example of Matthews, who was one of the proponents of this theory. Arguments for center of origin for mammalian species, where consecutive waves, and this is sort of the crux of it, consecutive waves of more advanced forms evolved and then displaced more primitive forms over time. And it sounds logical, like, oh, a new species evolves, somewhere, and then it disperses out from that center and evolves more, and things evolve more as they move out, and so you get this center origin and circular sort of dispersal out from that. It sounds good, but it's not always the case in how things work. This particular map is actually pretty problematic. I mean, this is one of the early forms of, they thought, like how humans may have evolved, and there was this idea of sort of northern superiority, where organisms that evolved in the northern hemisphere would sort of supplant organisms from the southern portions as they moved south. And obviously, when you apply this to a human context, it makes it pretty easy to justify racism and things like that. So this is problematic. But even beyond that, when you're looking at other types of organisms, this theory doesn't necessarily always hold up. And here we're looking here at some criteria that has been frequently used historically to identify a group, a taxon's center of origin. So basically you're looking for that center of origin, and there's a lot of different ways that people researchers used to basically identify that. So you could use the location of the greatest differentiation of type. So basically the greatest number of species. So where's the most biodiversity? You could look at where's the most abundance, like where are the most individuals. You could look at how closely related things are spatially. You could look at the size of the individuals, the primary productivity. If you're looking at something like vegetation, you can look at dispersal lines, restricted habitat. There's all sorts of things you could look at, like how organisms move around and those patterns. And really, there's all sorts of things that you could look at. And the problem here is that some of these may indicate a center of origin, and some of them may not. Individually, they're not very useful, because lots of interesting things and lots of weird things can happen over time, and organisms can shift and move around, and we've seen a lot of that. So if you're using, and I think this was probably the problem, is that people would use one or just one or two of these and basically say, okay, here's where most of the species are, so this must be the center of origin. And that's usually not, it's usually not the case. Like, not at any individual one of these is going to necessarily indicate that that's where a species originated or a group of species originated. So if you use a number of them, and they're all pointing to the same location, then maybe that's some pretty strong evidence. But I think the problem was that people sort of took this idea and ran with it, and would use any one of these and say, oh, here's the center of origin, and here's how this group of species evolved over time. So when you do that, you're not being very rigorous, and it becomes kind of like storytelling at that point rather than sort of a scientific explanation. And a great example of this is the sea snakes. So there's a group of sea snakes, and they're located mostly here, and they were originally thought to have originated in this area. So what you're looking at here is the range, and then the numbers indicate the species diversity for this family, I think, of sea snakes. So the number indicates how many individual species are found. Species of sea snakes are found in that location, and basically people thought that the highest biodiversity or species richness was found in this area right here, and so this is where they were thought to have originated and thought to have evolved from nearby terrestrial snake lineages, probably in Australia. And so they thought that if they looked at the phylogenies of these, they would be really closely related to those specific snakes lineages that they thought were to have evolved from, and then made their way um, into the water and became aquatic species. And so they thought this was the center, and then they sort of radiated out from that. So this would be like the oldest lineage. And that was what people thought for a while, until some more robust sort of DNA molecular phylogenetics came about. And so that's what we're looking at here. So here it's a multi-locus molecular phylogeny for the true sea snakes, which is this group here, and it's this group here. And basically, I'm not going to walk through all of this because, it's just a lot going on here. But they found that these species here were more distantly related than what they were originally thought to have been. And so it's sort of reshuffling where these lineages may have diverged from, and what some of their common ancestors were. And so it might not likely be the case that they evolved the way that people thought they did. And there's other evidence of that too, where there's a group that was supposed to be very closely related to this sea snake group is not that closely related. So it's still a little bit up for debate whether, excuse me, where exactly the center of origin for this species is. But it might not be exactly where people thought it was, just based off of the species richness. And so I think there's still some debate, but basically the more evidence that people are finding, it's sort of reshuffling the phylogenetic trees of these organisms. And so that just sort of highlights the problem with relying on those things like, oh, here's where the most species richness is, or whatever it is not necessarily going to indicate that is the center of origin, and that you can base your phylogenetic sort of evolutionary trees off of that alone. So that was center origin theory, and it was one of the earlier ones, and it sort of people sort of ran wild with it and used a lot of sort of, what do I want to say here? They kind of used it to justify a lot of science that wasn't very sound. And there was a big sort of pushback after a lot of that type of science was published. A lot of other people were sort of looking at this and saying, okay, we're not really being scientific here. We're sort of like fitting stories to our data rather than using the data to test hypotheses. And so in response to that, this guy named Crozat and a number of others, but he was the main guy, constructed this new or created this alternative theory called pan-biogeography. And this is done by plotting the distributions of endemic species and then drawing connecting lines between areas that share disjunct endemics to show areas of close relation. So basically what he did here was like taking maps, individual maps, and drawing circles around areas that have endemics and drawing lines that connect areas that have like sort of disjunct populations of different endemics. And then if you overlay those maps on top of each other and then sort of like statistically or even visually line some of these up where there's many connecting lines, then you can draw some inferences. And it's kind of a weird, different way of doing things. It does reveal some connections in some places, but it also connects places in others. Like here, like there's lines connecting northwestern North America to like Asia and like all the way all the way over here into Europe and stuff. So some weird things can happen here that may not actually reflect anything accurate in biogeographic history. But basically, this was a pushback to the center origin people who were basically the criticism of the center origin theory was that it wasn't being rigorous. It was just sort of coming up with stories that might fit the data. And so here it's more just about let's just use the data and see what those connections are. And it's an interesting sort of like it didn't really take on. Like some people thought it was interesting, but it kind of died off pretty quickly, except for a couple areas in New Zealand, a couple research groups in New Zealand, and I think in Latin America use this. And it's what's interesting about pan-biogeography is it's actually starting to see a reemergence in the scientific field where people are looking at this and saying, okay, maybe if we mathematically quantify some of these connections, we can get some idea, some statistical confidence about whether they're actually meaningful or not. So it's interesting in that it rose up in response to the center origin sort of lack of rigorous science, but people didn't really, this was so radical and so different that it didn't really take on very quickly, but it's interestingly now starting to see a reemergence. So it's sort of a reemerging newer area of biogeography. The area of biogeography or the approach that did become more prominent was the Hennig sort of phylogenetic approach here, or cladistic approach that we've actually talked about already. And so this was an early example of that, where this research group by this guy Brunden did an analysis of these midges of southern temperate forests and revealed that the forest biota of Australia had a closer historical relation to South America than New Zealand. So they're just looking at the phylogenies of these these midges of temperate forests. And what they found is that if you look, so here's basically we're seeing the genus of these or genera of these in the given region. So NZ would be New Zealand and Australia and South America here. And basically what you're seeing is that there's an earlier split from New Zealand and South America. So there's much closer relationships between Australia and South America than there is Australia and New Zealand. And if we look at the map, basically showing those dispersal areas, there were some major dispersal from South America to Antarctica over to Australia, but not as much dispersal from Australia to New Zealand. But there was a lot from New Zealand to Antarctica to South America. So basically, you're seeing those tracks of dispersal there, and this is one of the earliest uses of this sort of cladistic approach, where you're plotting out derived and shared characteristics and and the emergence of certain traits. And so we talked about that last week and saw how sort of you construct a cladogram using that kind of analysis. This was one of the first examples of that. Before we go any further, I kind of want to introduce this idea of a taxon area cladogram, because it's something we use to sort of tease out some of these scenarios. So we've already seen a taxon cladogram where it's basically constructed off of a trait or character state. So you have, an outgroup that may have some or doesn't have some characteristic, and then some characteristic evolves, and subsequent organisms, excuse me, they have those characteristics, and that's basically how you differentiate groups from one another, is the state of some character. You can also add the area onto these and get an idea of when certain areas separated from one another. So if we added, so here we have the taxon. Instead of showing the character trait, we're just showing the region of where these organisms are found. And so they're found in region A's, B, and C for organisms 3. 3 is in C, 2 is in B, and 1 is in region A. And these regions separated from one another in a similar sort of fashion, usually over geologic time, where things are drifting apart or some barrier formed or whatever it was. And the reason we make these is so that we can tease apart some different scenarios. And just to sort of illustrate that, we could have a dispersal scenario where an organism shows up in area A, disperses to B, and then to C. Or you could have the reverse. A shows up in area A and then disperses to C and then B. And both of these, would correspond to this single area cladogram. If you have vicariance, though, which again, just to remind you, is when you have a barrier that forms. You can have basically all organisms existing in areas A, B, and C, and then you could have a barrier form between A and B and C, and then you could have another barrier forming between B and C here, and this would result in this taxon area cladogram. So basically, you have two different dispersal hypotheses that could create the same area cladogram, but only one vicariance hypothesis that would result in that. And the key here is indicating that areas B and C are more closely related to each other than either of them are to area A. And we can actually take this even further. We could have the same taxon area cladogram where you have area A, B, and C, and let's just look at different dispersal scenarios. So you could have A, B, and C, and maybe you had a really straightforward scenario where you had one organism colonize here, and then disperse here, and then disperse here, and so you end up with this cladogram. You could have a different scenario where you have an organism colonize here, disperse here, then the original population goes extinct here, but then it is recolonized from over here, and then it goes extinct here, but it's recolonized here, and you can see it gets pretty tricky to keep track of all this, but it would result in the same area cladogram where B and C are more closely related to each other than they are to A. You could have the same thing happening over here, where you have a dispersal or a colonization, initial colonization here, dispersal here. This population goes extinct, becomes recolonized or redisperses from here, and then colonizes over here, disperses over here. Same area cladogram would be accurate. And then you could again have something, an initial colonization event here, and then dispersing here, and then going back, and then going extinct, and then going here, and then go. So basically the point is that it can be really complex. Dispersal makes things really complex, but they can result in the same taxon area cladogram. So this is one of the reasons why when you're using this approach, that it tends to be the initial hypothesis tends to be vicariance, because vicariance is much easier to disprove. So if you want sort of a elegant hypothesis, and something called parsimony, where you're assuming the simplest explanation first and then sort of disproving that from there. You want to start with vicariance, because there's only one vicariance scenario that could create this taxon area cladogram, but there's a number of dispersal scenarios that could create it. And that's almost always the case, because dispersal can kind of erase its own um, evidence by, recolonization and extinction, and going and doing all this can create the same taxon area cladogram. So that's something to keep in mind. Okay, so I've been going on for a little bit here. I'm going to wrap up this video. There's a reading about pan biogeography and how it's sort of reemerged a little bit and how it's being used. So read through that and keep working your way through the module. I will see you in the next video.